As Raju said, I work mostly with freshwater fish. So I work only with freshwater fish, and I work mostly on European, North African, and fishes of the Middle East. I did a bit in Southeast Asia. And if you work on the taxonomy of fishes in this region, so the West Palearctics, you will more or less automatically meet my papers. And if you want to publish in taxonomic realm, I am your subject editor in Zootaxa in this region, and also in Ichthyological Exploration, and even in other journals. It's very likely that you will meet me as a reviewer. So it's worthwhile for everybody who wants to work in this region to see which approaches we follow, and what are the taxonomic concepts and best practices in describing freshwater fish in this region. And of course, the concepts and practices should be the same worldwide. So a bit about myself. I'm already 56, so an elderly scientist meanwhile. And I started very young with science in youth research competition about phylogeny of lizards. I worked a lot with conservation and conservation biology since my childhood, and this continues through all of my research career. I made my PhD in the National History Museum in Bonn at Museum König on the ecology of freshwater fish. I worked then long at the Leibniz Institute of Freshwater Ecology and Inland Fisheries in Berlin. So IGB, again on freshwater ecosystems and how do freshwater fish organize within these ecosystems. So taxonomy was always just an element of what I did over the years. And as you see from my publication list, I have several other interests. And to be a pure taxonomist and only describe new species or work in taxonomy, I would personally say this is a bit boring. So I moved then to IDIF and worked as the director of GeoBonn. IDIF is the German Center for Integrative Biodiversity Research. I worked as a director of GeoBonn, and I was in touch with lots of international bodies like the Convention of Biological Diversity, the IPBES, and many global players in the biodiversity realm. Later, I worked again in IGB, or more on urban waters and German biodiversity. And now I am at the National History Museum in Berlin, where we have created a new center, which is the Center of Integrative Biodiversity Discovery, where we aim to increase the number of described species by developing new methods and new approaches. And you see, this is lots of things are going on, and there's lots of development also to improve this, the quality of species descriptions and to speed up species descriptions of all taxa groups, not only fish, and worldwide. So when I was always curious about freshwater fish, and I love to work with them. But when I worked in the ecology of freshwater fish, I learned there is many undescribed fish, and I found it's not so easy. It's easy to read the papers, but it's not so easy to write them. And there is somehow thousands of small details you have to consider. Every paper, every description you read, there is some problems, some challenges, and there is so many questions which come up when you really work on this, that I deeply believe you need, if you start in this field, you need a mentor. You need somebody whom you trust and whom you can always ask even stupid questions. And this is not necessarily always the case in the scientific community, but usually if you are a young scientist, you should have a PhD supervisor who is your person of trust and who is a mentor. But in my case, I made my PhD in fish ecology, so I had no PhD supervisor in taxonomy. So my mentor was Maurice Kotler, and I'm very grateful to him for many years. He was always helping me with all the tricky questions, and still he is a great authority in the field of fish taxonomy, maybe one of the greatest. So let's talk about fish. First of all, what is special about fish? So in contrast to many terrestrial animals and of course plants, fish live on islands. Rivers and lakes are more or less closed units to most of the fish species. So the river catchment is the spatial unit we usually look at. It is very likely that a species of leucistin, of dace or loach or whatever, or which is in one tributary of one river, is the same as in another tributary. So they are in a way replicates. This is not a strict rule, but it is usually the case. So fish live on islands, and the river systems are the units. Then, second, fish are of conservation concern. People care often about the survival of fish. So describing fish species is not just an academic exercise, but important to bring them into the red list and into conservation projects. So describing fish is a serious exercise, because when they are in the red list, there will be projects, people work on this, money will be spent, and so on and so on. And it's not so easy to say, oh yeah, this year we believe this is a different species, and next year say, oh, let's revise it, we do not longer believe this. We should be really very careful, and because it can have, there is a whole body of following actions in such a species description, what might be different from describing, let's say, some types of flies or small wasps or whatever, where there is no real conservation concern. Fish are a relatively small group. There are actually about 34,000 species of fish, 80,000 in freshwaters. This is in some insect groups, these are minor groups of species. But in fish, most species are described. So at least in my region, I would say 80 to 90% of the species are described. This might be different from other regions in the world, but usually half of the fish species are described. The types, so the holotypes and paratypes of fish, which are usually based in natural history museums or in collections, are not easy to access. This is not specific for fish, but we feel this challenge more and more. More and more taxonomists create private collections, more and more countries following also the Nagoya Protocol and other ABS mechanisms restrict or close the exchange of materials. It's more and more difficult to ship something and get it in loan. So types are more and more difficult to access. On the other hand, everybody almost can now produce a CO1 sequence, and we see a boom in availability of sequences. Many describers of species poorly read English and do not know the species concepts. So we have really bizarre philosophies of some describers, which I don't want to talk in detail here. But if we describe new species, of course, we need to read the concepts, what species are. I mean, this is like you describe a tree, but you have no idea how a tree is defined. So my talk is mostly about concepts. So let's talk about concepts and how do we approach the challenge. So in describing species, of course, you must be an expert in species concepts and in the process of speciations. Species are not clades in a tree. Species evolve and they are the result of speciation. And there is a lot of scientific research going on in the field of speciation research. How does it work and how does it function? Species are not the result of creation. DNA was not made by God to allow taxonomists to recognize species. This might seem trivial to most of you, but might come as a surprise to some of you. So species are not the result of creation, and they are not fixed units. They evolve and they speciate, and you have to understand the processes and the theories and read the papers which are behind all of that. This is really essential before you can work in this field. So let's have a look at the most common species concepts. There is books about species concepts, and we could make a whole week seminar only about species concepts. It's a very slippery floor, and I can touch this only at the surface. And of course, if you work in this field or want to work in this field, I really expect from you that you look deeply into literature. So species are made of individuals which look different and have different character states. And there is the typological species concept, for example, the only one we use in paleontology, which look at these character states. And you see the first line of species, a Ballitorid, Saccharus, Sevilla, a cichlid, and a cave cyprinid. And you see there's very different character states. Let's say the number of unbranched pelvic fin rays is very different in the first. The number of unbranched dorsal fin rays very different in the second, and the absence of an eye is very different in the third. Or so the type we find easy character states to distinguish these three. But look at the other three of them. We have clearly two groups. The left ones have a spotted color pattern. The right ones have a barred color pattern. So we also can easily recognize typologically two species. But of course, we understand that the left are the females of the right. So the typological species concept is, some people say, this is the species concept we use every day, and maybe it's correct. But others say this is a species concept in its pure form only used by paleontologists. And maybe this is correct also, because in the female-male question, we have additional information. We understand, in this genus always males and females look different. This is trivial. So the typological species concept has in the past often failed in this female-male question or a juvenile adult question. And still we have challenges today, for example, in surface and cave fishes, which might look very different. Or so the typological species concept would put these into two different species, these two garas here. But if we go to the evolutionary species concept later, we will discover that this is not correct. Then everybody knows the biological species concept. This is a species concept used or which you learn in school. Basically, this species, in the biological species concept, species are made of individuals which are reproductively isolated from other individuals in nature. So if we have a roach left, Rotulus, and a bream, right, Abrames, and we hybridize, and they hybridize what they do all the time in nature, you have hybrids, and if you back cross the hybrids, they should be sterile. This is very nice. And of course, if the hybrids in such a case are sterile, the parental species, the parental populations are two different species. But what is if it's the opposite? And we know that even very distantly related species can be hybridized, like the cat and the serval, and the hybrids like the savanna are at least partly fertile. Or you can hybridize a cow and or a bison, and there is a hybrid, the zubon, and this hybrid zubon is fertile. And even hybridization between cattle and bison happened naturally. So hybrid sterility, if hybrids are sterile, this is very nice character to say this is the parentals are different species, but if the hybrids are not sterile but fertile, this character completely fails. Then you know nothing. And we know that hybridization plays a major role in speciation, because speciation not just goes with selection. Hybridization can be a strong driver in speciation. We know, for example, these heliconus butterflies, two species hybridized and produced an independent third line, which is a species of hybrid origin. And even in freshwater fish, we know this as the Saroterodon cichlids from West Africa, where two Saroterodon hybridized and produced a third species of hybrid origin, which is well distinguished and is an own independent reproductive unit in nature. So this biological species concept, the hybridization test, the fertility test in the biological species concept is nice if the hybrids are sterile, but if not, it is useless. So hybrids, we have hybrids all the time, but as long as the parental species keep their identity in sympatry in nature, the biological species concept is fulfilled. And it is really reproductively isolated in nature. So you cannot just take them out, strip them, and check if the offsprings are fertile or not. So the biological species concept is applicable only in sympatric species. So if they live together in one place and keep their identity despite of certain hybridization, they are good species following the biological species concept. And this can be the case for very, very young species, but the fertility test, what we did in the 80s and so on, is more or less useless. But the biological species concept does not help you if the populations are allopatric. So what do we do if we have, and this is usually the case, if different species or population or we want to work about live in allopatry. So how many species do we have here? Is the upper one, this one, different? Is this one different, or is this one different? So all of them are allopatric, and only from this we have no way to decide it. The typological species concept would of course say that cave one is different, but indeed we know that this one is different, while this and this is the same. So if we want to deal with allopatric species, we usually apply the phylogenetic species concept. So the phylogeny reflects the evolutionary history of the species or of the fishes. The species are then this distinct cluster of organisms, and there must be an evidence from ancestry and descent. And this is, I guess, the standard today, that we apply molecular phylogenetic trees to go for the phylogenetic species concept. And it can look very nice, like in these persets, where all of the species which were usually recognized or since many years are represented here or in different clades of this tree. So the species, phylogenetic species concept defines species by their position and distinctiveness in trees, usually now in CO1 trees, but you can use very different markers, of course. What means just measuring the time passed since the last gene exchange. And this is something really important to remember. So CO1 or cytochrome B, which are often used markers, are not speciation genes. So a difference in these genes tells you nothing if there are species, because the characters which change, let's say, due to evolutionary pressures and which lead to speciation are very, very unlikely to be triggered by this cytochrome B or cytochrome 1 or cytochrome B genes. So the sequence differences in these markers is only reflects only the time spent since the last contact. Basically, they measure a kind of molecular clock and nothing more. Allopatric speciation is often associated with the time of isolation. So deep differences in CO1, so means long time since these populations have had no impact, no contact. And as the more time leads to more mutation and theoretically to more divergence, or this correlates with the, can be measured by the CO1 gene. But it's important to remember they reflect only the time, not speciation or adaptation or anything else. So speciation must not be associated with the time. And there is recently a very nice paper, not so recently, or 7 Deadly Sins in DNA Barcoding, and it's really worthwhile to read this to understand some of the pitfalls or in the DNA barcoding realm. So in some species, there are large molecular differences. And you see this here in Barbatula loaches, more or less, they're all identified as Barbatula sp, and there might be very many species involved. In other, often very young species, or there is no difference at all in the CO1 gene. Basically, they have all the same haplotype, while they, in this case, in the corygonids, they are often good biological species. So they live in sympatry, they keep their identity in nature. So it's not so easy. It's not so easy to say, oh, there is no difference in the CO1 gene, this must all be one species. Or they can be species which are very young and have not yet had the time to separate much deeper in these genetic markers. But we have many examples that speciation can be very fast within few thousand years, which you can, of course, then not measure with these markers, but you can use others where you can measure. So what to do? All depends on the species concept. The first example, the persets, no problem. The morphology and the molecular markers tell you the same story. The second example, the loaches, there might be several undescribed lineages which might be species. but they are not necessarily species only because they make different clades in a tree. And the third example, the whitefish, very young species might not yet detectable by the markers we have applied here. The critical question for many users is, are easy to distinguish species better than difficult to distinguish species? In this case, all the loaches would be the same. Or are old species better than young species? In this case, all the whitefish would be the same. What means then we have reproductively isolated, morphologically different populations in sympatry. And in the other, we have, in the loaches, we would have deeply molecularly separated allopatric species. Yeah, so you see a bit the tricks here. Before DNA technologies were invented and before everybody could apply them, we worked already in a way of integrative or iterative taxonomy. So if we would have two garas here, for example, or we would look at independent characters, ideally more than one, not be plausible to be determined by environmental factors. For example, these cave fishes are often determined by environmental factors. So one very clear or more nice characters, this was always the aim to find. And this, of course, is subjective and much depend on your experience. So for example, the habitat could influence the body depth, or fins could have different lengths, and so on and so on. So this needs a lot of experience. But before we searched for morphological characters independent from each other, and maybe formulate a hypothesis on one character and test it with another. In the last 20 years ago, I would say, an increasing body of molecular methodologies has been developed. We work since 10 years with CO1. We had a nationally funded project to barcode all the freshwater fishes of Europe, North Africa, and the Middle East. And we have, meanwhile, a barcode library of more than 90% of all the freshwater fishes of the West Palearctics. So basically, we have many more species than described in our barcoding library, and really almost all of them perfectly identified from type localities and so on. And from this, of course, we can derive relatively easy, if we have something new, a molecular hypothesis to be tested then by morphological characters. We usually apply the concept of iterative taxonomies. So again, species boundaries as hypotheses to be tested with independent evidence. But now we do not use different morphological characters of independent evidence, but we go to the field. Maybe we have a morphological hypothesis already in the field, but we just sequence everything through the for the barcode. We look at the tree, and then we check the different clusters if they can also be diagnosed by morphological characters. So if the molecular and morphological species hypothesis fit together, then everything is easy. And of course, we usually hope that this is the case. But in the last minutes, I will tell you some stories where this is not the case, because obviously this is the pepper in the soup of our work. The most easy situation and most astonishing situation to many newbies in this field are fishes which are often in sympatry, look very different. But if you sequence them, so we search for biological evidence, if we search for biological and evolutionary exploration for discordant evidence to distinguish between competing species boundaries hypotheses. So of mtDNA integration, usually in sympatric species or species living adjacent to each other, is not always easy to detect, especially if the fishes are closely related or and so on. We will publish soon a paper about some loaches from the Caucasus where we have two species involved. Here is Oxynomycilus elze, and this is Oxynomycilus branti from the Aras, and this is Oxynomycilus branti from the Kura. And of course, the working hypothesis was that this is 3 species. But this branti and this branti are by all means in morphology the same. We find no difference. While elze, we find nice differences to distinct, not super nice, but good enough to distinguish them from the 2 brantis. Oops. And so we suspect that also in this case, mtDNA integration play a role, but it would be needed to be tested in the future. There is many cases of recent speciation. I already mentioned this. Young species in whitefish are a challenge if you use these standard markers. We know that there are different species because they fulfill the biological species concept as they occur in sympatry. And naturally, with microsatellites, AFLPs, and so on, you can find that there are different reproductive units. But these cases also might happen in allopatry. So evolution can be very fast also in allopatry, and we might recognize very young species also in allopatry. So we see no major challenge in the concept of young species, also in allopatry, like in trouts, where this is a notorious phenomenon. But technically, they are not easy to approach, and the differences between populations and species, of course, remains challenging because this is clearly a continuum. So we still accept the morphological species, or even if they might be young. So like in this Albornoz, we have here in the box, we have 2 different species, or which are nested in between each other. But from the morphological point of view, they are really very different, and clearly if we can identify them. So we have not a problem with young species, even if they are not distinguished by the CO1 marker. A challenge remains with so-called cryptic species, or several authors we see in the last years. They just look at the tree, and if the fishes form a clade, they go ahead to describe these, even if the molecular distances, usually as the K2P distance, are very low. Or to say, here there's the barcoding gap, and so. And in this group of terrible Oxynomycilus loaches, we have 10 such clusters, and 4 of them have no names, but from a morphological point of view, they are more or less all the same. So very limited differences. And at the end, we see this is the red line of 2%. Most of them here to the right are not different more than 2% in the KP2 distances. So with KP2, we have only, above 3, we have 2%, we have only 3 groups and not 10. So what to do if the molecular species hypothesis is not supported by morphology? My best advice, also to myself very often, is work with other fish. Wait forever until more data are available. And we have already quite a bulk of species which where the molecular species hypothesis is not supported by morphology, and we have no very clear idea. We could recognize them as cryptic species, but the term cryptic species is very poorly defined. So we do not, it's not so, yeah, you know, this is a vague thing. And we could recognize groups above 2% as cryptic species, or above 10% or 3% or whatever. We must be very careful, because species are not just groups of animals which have a molecular distance in CO1. So since many years, there's a discussion about DNA taxonomy, and this could be a solution. And basically by this, we would replace all the species concept just by a threshold. So as still, I mean, some people work against this, still molecular data are freely available on GeneBank, and the open access of these data would allow easy species description if the species would be just based on molecules. So we could easily download everything and sequence another one and diagnose them on the molecules, or nobody would look into morphology anymore. And this is anyhow also because you have so many problems to get the to see the types, anyhow nasty. And why we should use 2%? There's no reason. We could describe species or with 1.5, with 1, 0.5, there would be more. And if morphological characters anyhow don't matter, this would lead to an inflation of morphological indistinguishable species, or which are just distinguished by minor molecular characters. And this is something what I see strongly as an editor. More and more people just sequence something and there's a minimum difference, and they say, oh, so many cryptic species exist, and they want to describe this. So we could make a rule and say all morphological species below 2% would be rejected, or everything above 2% would be species, everything would be so more easy, and the whole field is easy to go. Super. What do we do as a best practice? So at the same K2P distance, we usually describe, we treat two populations as one species if there are no morphological differences. So if the molecular distance is small and the morphological distance is also small, then they are one species, no discussion. We would describe the two populations with the same molecular distance as two species if there would be a nice morphological difference. So see these two Anatolichtis on the bottom of this slide, they are distinguished only by 0.7%. But this difference in the color pattern is really strong, and you can breed them and this stays constant and so on and so on. So we accepted these as two young species while the molecular distance is only 0.7. We learn more and more from more and more studies that there is no fixed threshold. So 2%, 1%, 0.3%, whatever. There is a continuum between populations and species. And these thresholds can help to look at this. But at the end, I would always avoid describing species with very small and unclear, whatever this might mean, depending on the group, morphological differences and low molecular support. So be careful, use your brain in this frame. We also did a lot with this automatic barcode gap identifiers, PTP and MPTP and so, but I must say I did not find these very helpful. If we are, let's say, in the gray zone between populations and species, it might even happen that you have a morphologically or homogeneous species which make different clades, and one of these clades might be morphologically different. So all of them are the same, but one group is different, what would make then the others paraphyletic. So you would need to describe several new species, which you cannot distinguish by morphology, only to recognize one group here, which is morphologically different. So should we describe species only to not to make them paraphyletic? Maybe not. But this is an ongoing discussion, and there might be very many different opinions. And I hope there's very many different opinions on all what I say here. So you will see I come to the end of my long talk, and I say it a bit with this little lady. So I learned, and also myself, I learned a little bit about the topic. So I'm going to read academic papers to learn more. And that now I have learned that I know nothing about the topic. And the solution is to be active and to learn more and read more and talk with people and try to get a theoretical backbone and a good idea. There is many publications in this field, and of course I cannot tell you everything here, and I don't want. There is books about how to describe species, and there is excellent papers like this book also, Do Species Exist at All? Or read some things about species concepts. And if you want to work with us, actually until the 16th of April, there is a synthesis call open for which you can apply to visit our museum, hopefully when Corona will allow this. And as I said before, I'm very happy to discuss with you. So some warm words which you might allow me as an elderly scientist. If you start, you will make mistakes and you must have the backbone to accept them. Maybe synonymize your own species if new data come in. So it's not a holy cow. Read carefully the rules and formats of the journals. As an editor, I can tell you, this is the pain at all. People submit papers and they didn't care about the formats of the journal at all. Check at previous papers how they were done. Your manuscript will be read, but you must see this positive. Somebody took the time to read all of this and think about. And it happened to me also at the very beginning. It was a real pain. My manuscripts were in a terrible shape when they came back from reviewers, but you have to see this as a challenge and positive. Accept criticism, but you of course should have an own opinion, and you can always ask an editor to rule. And never just send rejected papers to other journals without taking as much advice from reviewers as possible. I can tell you, this world is a small community, and we will, when I had the case to see a paper unchanged 3 times from different journals as a reviewer, and this will not shed a good light on you. So all reviewers and editors and all colleagues are there to help and not just to reject you. And I thank you very much for your attention. This was a very long talk, and I hope you pick something interesting for your future work from this.